Hello everyone, welcome. So glad you can join us. I'm Phillip Compeau. If you've joined us for one of our streams in the past, good to see you again. I've seen a couple of, looks like new names and faces in the chat, so welcome if this is your first live stream with us. I would say, especially if it is, feel free to just say hello in the chat. Be good to see some new folks here. We're going to be solving a collection of Rosalind problems, maybe all of them in our bioinformatics stronghold. We'll see how popular this is. Maybe a big chunk of them. And today we're going to start with some of the introductory ones in the bioinformatics stronghold. So there's about 100 problems on Rosalind in this collection of questions that are inspired by challenges in bioinformatics. And we can see that there's quite a lot to do. You also see what's nice about Rosalind is it shows you the number of people who have completed things. So can we become one of the 100 people or so who have actually gone through every single challenge? That's going to require quite a bit of work. So we'll see. Today we're going to solve, I think, 8 challenges, and I have them sort of at the top in additional tabs. So we're going to go through and solve what the first 8 questions are. And what's nice about Rosalind is we also have a tree organizing all the problems. So you have to solve this question before you can unlock this question, before you can unlock this question, just to make sure that you're sort of on the right track with some of our initial questions that we're looking at. And so it's neat because the platform produces this network of sorts where you can go down through different layers. And the idea is sometimes you'll get a question that's a little easier than what you have seen. But for the most part, the questions become progressively more difficult as you get farther down in this tree or in this network, where all of the arrows into a problem indicate problems that you have to solve before you encounter this. So we'll see how far we go. For today, we're just going to try and get to this layer. We're going to do everybody in this layer except for this question, which is a genetics question, which is a little different to all the other questions that we're going to look at today. All these other questions, though, are all about string algorithms. So we're looking at, I think, relatively straightforward questions about things like DNA strings or RNA strings or proteins, questions that we might want to answer about them, that we can write a little bit of code to do so. And I should say, this is, in many senses, this is essentially a love letter to this Rosalind platform that I had the privilege of co-founding about 15 years ago. And before I do any coding, I should give a special thanks to all the team of folks who helped build this out and worked immensely hard on making something that, at that point in time, our goal was to reach 1,000 people. We thought if we could ever make an independent learning platform that people could adopt in university courses, but that also could be this resource for independent learning. If we could reach 1,000 people, we felt like we were doing a good job. And we had 1,000 people in a few months. And since then, it's nice to see nearly 130,000 people have solved at least one question on this. And we've seen it adopted in, like, I want to say around 500 universities have used this. So that has honestly been an honor, and a thanks go out to the entire team that we worked with to make this. Special thanks, I should say, because I wrote, I want to say, essentially every question in the bioinformatics stronghold. There might have been a couple, I think, that were like community driven that we then edited. But special thanks to Alexei Kladoff, who was our wizard programmer who helped put together all of the autograders and so forth behind the scenes. And I worked closely with during that time. And I was really proud of what we were able to achieve, because we're able to structure all these problems in this giant network with one question kind of building on the other. It was a huge amount of work, but very much worth it. And he was absolutely instrumental in doing this. So my dark secret is I don't think I'm one of the people who has solved all 105 questions here in the stronghold myself. I've solved quite a few of them, but I've wanted to take the time to just go through these questions and write code to solve all of them. So there's a little bit of a guilty pleasure for me in having the chance to go through this and do it, because it will give me an opportunity to just practice this. One important consideration is, I think our website here is nostalgic, let's say. Should we say nostalgic? It's of its era, and I think that's a good thing in a certain sense. And I also think there are a lot of people who might say, well, you're solving questions with code, and meanwhile, AI bots can write code better than most humans can very, very quickly. And what's the value in doing something like this? And what I want to focus on with this is, yes, code can be generated very quickly, but what we're ultimately doing is not so much coding as solving problems and thinking through how we approach the problem-solving process. So that's how I want to first and foremost frame what we're going to be doing is, yes, we're going to go through these and see some code, but I think if you've learned with me before, there's a much higher principle here that we're trying to get to, which is the notion of how do we take a problem and break it up into pieces and figure out exactly like what the best tactics are of solving that problem. And we're going to use Python to do that. And the other thing I should say is, even though we're going to be coding in Python, we're going to see some things that are not specific to Python necessarily. So I'm going to solve problems in ways that I think are very portable across languages. One of the things about Python is there are a lot of solutions that might be very specific to the language that I could show you here that might make things very simple, but I don't think are necessarily very transferable in terms of problem solving, or very transferable in the case of that you would solve a question in another language. So that's what we're going to do. We also have, if you're new, we have a programming course that's kind of running concurrently to this, and you're welcome to join us for that. So we're running a course, I would say, synchronously on our streams, and the course is going to live asynchronously. We're making tons of updates to our website currently to add a lot more functionality to it, so that you can come to it like anytime you like and complete the curriculum that you see there and get a certificate and all this sort of stuff. So that's our central hub for our general introduction to programming. We're not going to start here because I'd say the level of even the first problem on Rosalind is probably around the middle of the second chapter of our programming course. So there is kind of a little bit of a starting point in this, and it was one of the things that I remember learning when we made the platform was these first three questions in the tree. I think the first questions that I was trying to design were kind of at this level or even deeper. And then I realized we needed some more introductory challenges, because if you're comfortable with programming, then you probably won't find these challenges that difficult, but you'll go through them quite quickly. If you're not, then these are good practice opportunities for you to get comfortable with what we're going to be doing. So our programming course lives at programmingforlovers.com if you're just interested in a general introduction to programming. Also, we've got tons of resources, and this is all in our description, and it's mentioned in this header that we have here. But join our Discord. That's a great place for where we build community. Join our mailing list to kind of stay in the loop about what we're doing, because we're not just doing this stream. We're not just launching a programming course. I have a course called Great Ideas in Computational Biology that we're launching very soon. That will be a more general approach to what are these ideas in the intersection of computer science and biology that have made biology into this data-driven field. And that's based off of teaching that I've done at Carnegie Mellon for the last 10 years. So the way to stay in the loop about all that is join our mailing list, hop in our Discord, all that good stuff. And I'll actually be launching something beyond just these courses. So if you join our mailing list, that's a great way to stay in the loop around some of the other exciting stuff that we're about to start doing. All right. Only other thing I would say is, before we get started here, like and subscribe if you haven't done so already. That helps us stay visible. If you're joining us live, please feel free to use the chat. It's great to see everybody. Thank you for joining us. If you're watching us on the recorded video on demand, just make sure to give us a comment so we know how we're doing. And yeah, I think we're ready to get started. So you'll notice that I should say we've got a little bit of setup. So what do you need to do? You need an account at rosalind.info. That would be helpful. And you also are going to need to install a couple things. You can use any text editor that you like for writing the code, or if you want to just follow along with what I'm doing, that's fine. But if you want to actually follow me as you go through this in real time, then you need a couple of things. And those two things in particular are a text editor, VS Code, and Python installed on your computer. One of the things that you'll know if you follow me for Programming for Lovers Python streams is that Python irks me. Python is kind of my nemesis. And so one of the reasons why is that I have found it's one of the hardest languages for learners to actually come to and install. There are a lot of languages that you literally just click download, click install, and then you're good to go. With Python, sometimes people have multiple installations, and this can be tricky. So I wanted to say, make sure that you check out our instructions for installing Python at programmingforlovers.com. It's like our first code along. If you have any challenges with getting Python installed, because this is a great way of just having a simple installation that's not built on a lot of other complicated stuff, and you can check it out there. So final point, if you've got Python installed and you have VS Code or a text editor that you're comfortable using, I've put some starter code or a link to a folder of starter code in the pinned chat. So if you see that, and I'll add another comment to it here live, I've placed a folder there that just contains a bunch of Python files of what we're going to work on today. We're going to fill those in. So when you go there, you're going to see a Rosalind folder. It's got a bunch of Python files, and you can download that and place that anywhere you like on your computer. And when you do that, I've opened that folder in Visual Studio Code. So when you open VS Code, you can just click and drag that folder, and my Rosalind folder comes up here, and I just put mine on the desktop. So this is just a desktop folder that's named Rosalind, and it's got all of the starter code, so you can see it's not filled in yet, and that's what we're going to do here. I've got a couple places where I've added a little bit of stuff that will be helpful to us as we go through this. The number one thing is that I've got printing already set up behind the scenes, and a lot of this is just the sample data sets that are provided on Rosalind. I just set this up already. And then our job is just to write the highest level function to solve the question. We'll talk about that here in a second. And anyway, this is what you're going to see. The first question that we're going to solve is this, is the first one alphabetically called DNA. And we're going to go through and look at each of these questions individually. But I think we're ready to get started. I'm going to point us to the specific web page here, which if you look at the problems list, it will show up as yellow if you have a new account, but it's the top one in the list, and it's the top one in the tree. So that's the only one that's really going to be unlocked for us, and that's where we should start. So we're going to get started here momentarily, and if you have any questions, let us know, and then I'm going to start with the first problem here in a second. Other thing I would say is let us know where you're from too if you're saying hi. Ontario, Cambridge, Mass. Welcome, welcome. So some folks in North America. Good to see some Europeans. If you've wanted to join us and you've not been in a time zone that's been amenable for our evening live streams, and this is your first one, great. Thank you for coming. We appreciate that. Awesome. Lovely. Ireland, Belgium, Germany, India. Great. Nicole, welcome from Yorkshire. All right. Welcome from Dubai. Hope you're doing okay. Tanzania, that's amazing. Welcome, Oscar. All right, it's great. It's great to see the turnout. So this is nice. I know we've, I do have a day job, so it has it's been easiest for us to have our streams in the evening, but I think this is a good sign. So we've got a lot of folks here. Wonderful to see you all. Thanks for joining us. I think it's a good sign that we should keep doing these at a time that's during the day. So keep coming. Hopefully this is helpful. Welcome, and I think we're ready to get started. So we're going to be solving the first question, which is this counting DNA nucleotides question. And if you look at the typical question that you see on Rosalind, there's always a collapsible introduction to the biological side of what we're doing. And in this particular case, this intro to the biology is just what's an overview of what is DNA. And I think probably if you're here, I probably don't need to explain that DNA is a double helix and that it's a bunch of A's, C's, G's, and T's. So I'm not going to go through the biological side of questions today. But the idea is each of these questions is inspired by that science in some fashion. And we're going to be working today exclusively with strings. And so what are strings? Strings are these ordered collections of symbols that are taken from some alphabet. And in the case of DNA, we have just a 4-symbol alphabet. Now we'll talk about proteins and peptides here in a little bit, where our alphabet changes from 4 symbols to 20 symbols because we have 20 standard amino acids that we typically use. And in this particular case, that we're looking at a DNA string of length at most 1,000 nucleotides, and that's what I'm going to be given. And we're going to know that we're in this 4-symbol alphabet. And what I want to do is produce an output that corresponds to the number of A's, C's, G's, and T's that we see in that string. And so from a human perspective, it's not a hard question. You can imagine what we need to do is just range through the string and count up the number of symbols that we see. The question is how to take this and put it into code. And so what I will do is I'm going to look at my starter code that I have in VS Code. And what I want to write, and you'll notice, I should go back here, we have our sample data set. And how I'm going to solve the questions is to make sure that our code is working on the sample data set that we see. So we will have our input, and I've got a little bit of code in main that already has that specific input from the site. So this allows us to check our work and ensure that what we're seeing is matching what we see on the website. And then we're going to go through the process of actually submitting our solution. So how Rosalind works, if you're not familiar with it, is that when I click download dataset, it's going to take me to a randomized data set, and then I'm going to have a standard amount of time to upload my answer to this. So what I should do then is write a function. What we're going to see is, in general, I could write all my code in main, but the first piece of transferable knowledge, that is something that if you follow our programming course, you're going to have seen me talk about quite a bit, is the idea of thinking about when you have the problem that you're interested in solving, that has an input and it has an output associated with it. And that means that we should have a function at the highest level that solves that question. And in our case, that function is going to be called count nucleotides. So we have the def keyword to indicate that we've got a function that we're defining. And it's going to take a string s as input. And one of the ways that I always try to write Python code is to make sure that I do two things stylistically. Number one, I'm going to think about everything in terms of the types of the variables that we have. So in this particular case, I'm working with a string, and I can specify to Python that I know that this needs to be a string. I can also think about what does this function need to return. And in this particular case, well, maybe that's not completely obvious to you yet, but we should never write any code unless it's completely clear to us on a high level what the input should be and what the output should be. And so if I weren't clear about that, I could sit and think about it, but I'm going to write what's called a doc string or a documentation string that is enclosed between these three double quotes that we see. So good Python practice is always to say, I want to count, say, on a high level, what is it that this function does? And what this function does is it counts the nucleotide occurrences of each nucleotide in a DNA string. And we're also going to specify what are the arguments, meaning what are the parameters or inputs that we have. And I'm going to say we have a string s, and it's not just a string, it's a DNA string that consists of the characters from the alphabet A, C, G, and T. We always also think about a function in terms of the output of that function. So I'm also going to have a returns, and here's where we think about what exactly is it that this function needs to return, right? So if I'm going to be returning something that corresponds to A's, C's, G's, and T's. Now, one of the things I could do is I could simply return like a list of 4 elements, one for the A, one for the C, one for the G, one for the T, but that's also a fixed length. So I could also return a tuple. So a tuple would just be, because I know that it only has 4 things in it, 4 integers. That would be nice. I think that there's a better way, though, which is to not lose the information that each of these numbers corresponds to a certain letter. And so how I'm going to return this is as a dictionary that maps each nucleotide to its count. And by its count, I say number of occurrences in S, okay? And what's nice about that is I've not written any code yet, right? But I've thought through the problem carefully without thinking about how I'm going to solve it. But I've done a lot of the work that's going to prevent me from starting to write code in the wrong direction and making sure that I'm clear about exactly what problem it is that I want to solve. When I teach students in person, I get the chance at Carnegie Mellon to teach a lot of smart students. But one of the things that I've realized is a huge opportunity for me is the notion of, and I think this is more relevant in an AI era than it's ever been, is that we can fall in love with algorithms and methods and these sorts of things. But a lot of times when you encounter something new or you haven't done something before, one of the critical abilities that you can have is to be able to frame carefully, what problem is it that we're solving exactly? All right. And so this is a relatively straightforward question, but I think we're still starting to see that, right? Taking a step back and saying, what is it that I'm solving precisely is going to be very helpful to us. And in this case, I think that helps us think about, well, what's the form that this data should take? And I'm going to specify this as the output of the function, which is a dictionary that maps strings to integers. Again, I've typed a lot. I've not really written any code, but now that I've understood exactly what this function should do, it's going to make it a lot easier to write. And you might not have seen Python written like this before, and if you haven't, that's cool. This is a relatively new development in Python to allow you to specify what are called type hints for the inputs and the outputs of the function. This is required by a lot of languages, and so this is an example of the type of thing I was talking about, which is the opportunity to show you something that is more general than just learning Python, so you're comfortable if you see it in another context. For now, though, I want to solve the question. And if I want to solve the question, well, the first thing I might do is to say, let's make a dictionary with our 4 keys. So we're going to have one for A, one for C, one for G, one for T. And they're going to have 0 values initially. So to do that, I'm going to say, let's set new counts. That's going to be what I call the dictionary. And a lot of times you'll see a blank dictionary or something like that, or we could go through and set the values as we go through. I'm actually going to do the same thing that I'm talking about before. I'm remembering that I'm mapping strings to integers, and Python lets me do this. So again, this is the sort of thing that's required in other languages. In Python, you have all this flexibility, which a lot of times for beginners can be a challenge. And I always suggest think through what this dictionary is going to be. So when I create the dictionary, I'm going to specify what its type is going to be. And by its type, I mean it's going to have keys that are strings and values that are integers. Once I've done that, then I'm just going to go through and say, okay, we start with a 0 value for all of the nucleotides that I'm going to consider. So I'm going to say A gets a value of 0, so does C, G, and then T2. The other way that I usually write code is to think about, well, this is the thing that I care about at the very end of solving this. Once I have this filled in with all the values, then I've essentially solved the problem. It's just a matter of printing it out in a nice way. So we're going to fill in our code here, but then in the meantime, I'm not going to worry about exactly what that code is going to be. I'm going to remember that at the end of this function, I want to return what it is that I care about. So I think about creating this data that isn't complete yet. And then at the end, I want to return the data. So that's what the function is going to produce. Then in the middle of this, we of course need to actually fill in our code. And what's nice is that Python provides a pretty straightforward way of ranging over a string. So if we remember, we've got our string here, and we want to go through all of the symbols in that string. And then we want to, as we encounter each individual symbol, so when I'm at this symbol, for example, I want to increase the count associated with that letter. So this would be the 3rd time I've seen a T. So when I'm at this particular letter, new counts of T is probably going to be 2 at this point, I hope. And then I'm just going to be adding 1 to it. So when we fill in our code, I'm going to say, what is it that I'm doing? We range over all the symbols of S, and for each new symbol that we encounter, add 1 to the associated value of it in the dictionary. So I can range over these symbols by saying for and then some variable name in S. And the variable that I'm going to call it is, well, we're working with DNA, right? So let's call this thing nucleotide. And now nucleotide is going to hold this current symbol. So as I range through S, the current symbol is going to be called this variable nucleotide. And now it's just a matter of saying, okay, so what is it? And the word, we also want to think about, well, what are the things that could cause us troubles, right? This is not going to be the case for Rosalind. It's always definitely going to generate a data set for you that contains A's, C's, G's, and T's. But if we focus on good programming style, one of the things that we might want to make sure of is, let's ensure that it's actually an A, C, G, or T. And we can have a simple check of that by saying, well, is it the case that the nucleotide that we're looking at is not a key of the dictionary? I could go in and manually check that it's A, a C, a G, or a T, and if it's not one of those, then that would be bad. But I can also just do a quick check to say, is it not a key of this thing? Because if the current letter is not a key of the dictionary, something has gone wrong. And I can make that check with, if nucleotide not in, what did I call it? Nuke counts, okay? Meaning current symbol is not a key of dictionary. And if that's the case, that's bad. So what I want to do is I probably want to do something like raise a value error and say in this case, error current symbol is not a DNA nucleotide letter. That's a nice quick check. It'll make your code stronger. And the point is, if I make it here, I know that the letter is an A, a C, a G, or a T. And now it's just a matter of incrementing the value associated with the current letter. So you can pick your favorite position. If this is the current symbol we're at, well, all I want to do is increase the value in the dictionary associated with the A that I see. So what I'm going to say is, we'll say nuke counts of my nucleotide, and the shorthand quick way of doing that is to say plus equal 1. Okay. When that happens, for the uninitiated, it's equivalent to this statement that it's equal to its current value plus 1. But that's such a common construct that we see in programming that pretty much every programming language these days is going to allow you to just say plus equal to 1 and increase the current value of that dictionary element by 1. Now the hope is that we've solved the question. I hope so. We're not sure. And we now need to check. So in order to check, well, I've got a little bit of code here that's set a specific string for us. And then we want to count the number of nucleotides in that string. And then we're going to print out the result. Okay. All right. So to do that, we're going to pull up this sort of bottom ribbon that we have. And when we have a terminal here, it's actually the nice thing about VS Code is that it's going to navigate me into my Rosalind folder that I have here. So if I type ls, for example, you're going to see here are all of the file names that we see in the folder. And once I've got all these file names, I can run each individual file that I see. So if I've got Python installed, all I have to do in order to run this file, which is dna.py, is to type Python 3, or if I'm on a Windows machine, it would just be Python. But I think Python 3 probably works for everyone. And then dna.py. So this is just a command line command that tells Python to run the code that is contained in dna.py. And here, this line of code says run what is in here. And what is in here is the creation of a string, and then calling our function and printing it. So when we run this, we'll see, hopefully, okay, here's our dictionary that got printed. And we can go in and check, well, A is 20, C is 12, G is 17, and T is 21. So when we look at what we see in this problem, it appears that we've solved the question. Now, maybe we haven't. Maybe we just solved it for this one specific question or one specific data set. So we would need to check this on a lot of different data sets. Rosalind's format is, if you can do it on a big randomized data set, then you probably have solved the question. And so that's what we need to do. The other issue that we have, though, is that, again, if we look back at the website, we have to format our output in this precise format. So we have to say 20, 12, 17, and 21. And that's different from how this dictionary has been printed to us. So we just need to make sure, and as we get towards more advanced questions, I'm going to write probably a little bit of code for us, or have starter code that already handles a lot of this output processing, because this is one of the least exciting parts of programming, to make sure that you're formatting a print statement in a way that like gives you the exact thing that you want. In this particular case, I'm not just going to print the counts. I'm going to print counts of A first, and then counts of C, and then counts of G, and then counts of T. So that when I run this thing, again, I can just hit the up arrow in my terminal, And when I run it, I see 20, 12, 17, 21. So now I feel a lot better or more confident that this is going to work because I've gotten the correct answer on this small data set, but it also matches what I see in terms of the formatting on the site. Let's try this then. So let's see how we do. We can click download data set. And behind the scenes, that's going to give me a .txt file. And that .txt file for this particular case gives me this data set. You'll get a different one if you're solving along with us. But I can copy that string for now. We're going to see a better way of doing this here soon. But for now, I'm going to copy my string. And I'm just going to place it into main. And make sure that I save my code. So meanwhile, when I look at Rosalind, the clock is ticking. This is something that I'm sure for a generation of folks solving our problems is probably initially a little anxiety inducing because the timer has started. And if I don't solve this within the allotted time, then I think there's like a minute timeout that I have to wait before I can get another data set. But what I want to show you is for this particular platform, this is why the idea is to make sure that I've written all of everything in advance and that everything is working on a sample data set. It appears to match the sample output because 5 minutes is plenty of time to run our code. Our code is going to run very quickly. So when I save this and I run my code again, now I get these 4 values of 205, 197, 207, and 220. And I'm just going to copy them back over into the output submission box. And I also could attach my file. I could provide my code too if I wanted to share my solution with other Rosalind users. I'm not going to take that direction. I'm just going to copy things in for now and click submit. And we solved the question. Excellent. So yay, we've joined 130,000 other people, or I guess in this question, for this question, 70-some thousand people who have solved this question. And we're doing good. All right. It's always good to make sure that we don't have a huge bug or something that I have to encounter on our first question out of the gate. In the meantime, so we're going to keep solving questions. In the meantime, how are we doing? Any questions, comments, thoughts from me? Anything I can clarify about the code that we just wrote? How are we doing? And Rodney, welcome to you from Kenya. One good question is there a faster way of writing our code? Is there a better way of writing this code that wouldn't necessarily have, I mean, I have a bunch of comments here, right? But that wouldn't necessarily have 20 lines associated with it. And that's a good question. And I think this gets at what I was talking about before, which is I am not going to focus heavily on the most Pythonic way to solve these questions. I'm using Python because people like Python. A lot of people know it. It's, it's the language of scientific programming. But there is not going to be a whole lot transferable if I write like a one-line zinger that is useful for this problem that then doesn't actually help us understand how to solve another problem when that specific way that is very precise to this specific thing that we're solving, when we see another problem where it's slightly different. And so one of my frustrations with trying to teach that is that, well, how is it that, what is the idea? Does that mean that to learn how to program and to learn Python, you're actually just memorizing a lot of shorthand stuff? And my sense is that kind of skill is increasingly dying because my solution is still very much a solution. And I would argue that it's way more teachable and understandable than a lot of the very tight Pythonic one-line ways of writing stuff. So this is something that you're going to see throughout. You may have better solutions than mine. Frankly, a lot of these one-line solutions are better. They might even be faster, but they're not the most teachable or transferable. And so that's why I don't focus on them. But it's a good point. All right. Other question. Are we going to see more of the thought process of thinking end-to-end? Yes, very much so. Every question I'm going to do the same thing. We might hit a point eventually here where, I don't know, I give you all of this because it's obvious at that point after we've solved 30 questions or something. I don't know. But for everything that we do today, I'm going to write the whole doc string. I'm going to write all of the type hints, and we're going to think about everything we do in terms of input and output, even when that might be kind of straightforward. Okay. All right, good. So let's do another one. Let's look at the second question that we have on the site. And the second problem that we have in the tree or in the problem list is going to be called our RNA ID, which is transcribing DNA into RNA. And the biological explanation here is, well, RNA is in many senses, a single-stranded version of DNA where thymine gets replaced with uracil, but there's still a 4-character alphabet. And the idea is that there's a hypothesis that maybe RNA was the first genetic material, and then DNA, because of its greater security as a molecule, it's not as susceptible to mutations, for example, because it's double-stranded and the genetic material is locked across the double helix with base pairing. It was an evolved form of RNA. But our cells still use RNA as a messenger for a lot of reasons, but in the greatest sense as a messenger to take the DNA blueprint information, to transcribe it, and then to send that out into the cell for the production of a protein. So we're going to talk a little bit about production of the protein from an RNA string shortly. But for now, there's a natural question to solve, which is that I have a DNA string of some length, and I want to produce an RNA string from it. And at the end of the day, if you look at the sample data set, you say, well, on a human level, this is a pretty straightforward question, right? I'm just replacing all of the T's with a U. On the level of code, we have to be a little bit careful here to make sure that we're taking that idea and we're translating it appropriately. And there's actually something to be learned about efficiency in this problem, even though it might not seem like a particularly challenging one. There's a transferable skill that applies to Python, but that applies to a lot of other languages as well. So I'm going to open up rna.py, and you'll see I've got a little code in main that has the sample DNA string that says I'm going to have another string resulting from transcribing that string, and then I'm going to print it. So I don't have to worry too much about my print statement and consistency for this particular problem. But the question or the function I want to write is going to be called transcribe. And it takes a DNA string and it produces an RNA string corresponding to it. So I'll say, before I get ahead of myself, we're going to have a doc string. And what does this do? It transcribes a DNA string into an RNA string by replacing T with U. So our arguments, we're going to take DNA, which is a DNA string from the alphabet A, C, G, and T. And then the return is this corresponding string that replaces T with U. Okay. And now I understand what I'm doing. I start with a string, I return a string, and I'm going to write my code by saying, well, the DNA string that's input is called DNA. The output string that I want to produce is going to be called RNA, and I'm going to start it off as an empty string, and then we're going to return it. Then in the meantime, I need to fill in my code. So again, I'm writing code in the same way. I think about everything in terms of types. I think about everything in terms of what is the output that I want, and I want to make sure that, a lot of times when you learn to code, it's easy to forget your return statement. And I'll acknowledge sometimes I do it too, typing along something and then you forget the return statement. If you have joined me before, you'll see that a lot of times I write code in this way. So what is it that we want? What is it that we're going to return? And I delay. I'm a lazy programmer, which means I'm better than I used to be, because laziness is a virtue, but we're going to delay what we're going to do in terms of the engine of the function for as long as possible. And now we can write that engine, of course, because I can say, I'm doing the same thing that I did before. I'm ranging over the string, and I'm looking at the current symbol. So I could call this symbol, I could call it nucleotide, either way. And I can make sure that I have an error check. Now here I'm not going to do an error check, but I point out that's a good thing to do. And I'm going to say if the symbol is equal to T, well, I want to add or concatenate the symbol U to my growing RNA string. So the idea is I'm ranging over the DNA string, and I'm building up an RNA string one symbol at a time. Most of the time, I'm going to keep the same symbol unless I encounter a T, in which case I want to append a U. So I'm going to have an else statement here, which says RNA plus equals the current symbol. The current symbol is a string, and so I'm just string concatenating one symbol at a time as we go. Then I get to the end of that and I return RNA. So our function's quite short, and I can run it and make sure everything is working. So in this particular case, I'm going to pull up my terminal, and I'm running rna.py. And I'll see I get this result. It appears to be everything's in order. The T's are getting replaced with U's, which is what we wanted. Good. The point is, this actually can be improved. And yes, one way that maybe it could be improved is I could write a really short, tight, one-line Pythonic thing. But that's not what I mean. Actually, this function, which is going to produce repeated string concatenations, and this is true not just of Python, but a lot of languages, is as we go through this, making, say we tried to do this for an entire chromosome. Well, you wouldn't do this for an entire chromosome, but let's say you do it for a long gene or something, tens of thousands of nucleotides. If you were to run this, you might actually see it starts to get slow. And the reason why is every time Python does this statement, it basically creates a new string, and it takes all of the first string and places it where in the start, and then it adds the single symbol U to it. So it's adding one symbol at a time, but it's doing that with a huge number of like copy operations. And so as a result, this doesn't scale very well. To make it scale very well, we're going to do something that is pretty common across languages, which is instead of creating a string, I'm going to create a characters list of strings. So it is an empty string called characters. And when I range over DNA, if the current symbol is T, instead of concatenating to a string, I'm going to append a U to it. And same thing here. Instead of concatenating strings, I'm going to append the appropriate symbol to the list. So as we go through, we're creating just a list of characters, and that's honestly not tremendously dissimilar to probably how you think about a string. A string is kind of a list of symbols, except that it's in Python it's immutable, which is going to cause this issue that we see here. So this is more of an advanced idea, but what it means is at the end of this process, I need to return a string, right? The last thing I want to do is return characters, because that's not right. I said I wanted to return a string. And to take this list of strings and convert it into a string, which will actually be fast, here's where I will use a little bit of Python knowledge to say, I'm going to join all the characters together, and here's one way of doing it. This tells you what to place in between. So this is the separator in between characters that you see. This is the empty string. So this just says join everything together, but don't put any space in between them. Okay, so I'll show you what I mean. Like say I added a space there. When we run this, notice that I don't get a string, or I do get a string, but there's a lot of spaces in between them. So I don't want any spaces between the characters. So when I save that and I run it, you'll see here's my string. So we should look at a larger data set. And when we look at a larger data set, we should go to Rosalind. It says I have not solved counting DNA nucleotides, so let me refresh the page and see that I have. I'm going to download my data set. It's going to give me a .txt. Open that. Here's my data set. And I'm going to copy that into my VS Code. And run, save. Hit the up arrow to run this thing again. And now I'm going to copy my string that I have, take it back to the platform, copy it in, submit, and hopefully I get it right. And I do. And we're getting all kinds of achievements. I think we've gotten the Phi X174 achievement. So this is, we're growing as an organism. So we're up to a bacterial phage. And hopefully as we solve more problems, we're going to get become a more sophisticated organism. It's one of my happier gamified parts of the website. And we've got 2 problems now. All right. Questions? Anything we can help with? We're going to start solving more complex ones. Is there a built-in replace function? Absolutely. Of course. Does it help you know how to solve a harder problem? Nah, not really. Okay, good. Let's see a more advanced question as we go. So, can I repeat what is the difference between a character and a string, Alexandra? Yeah, it depends on the language. In a lot of languages, you have a special type that's devoted to a single symbol. And in Python, you don't have a single symbol types. Everybody's just a string. So when I say a character, what I mean is a string of length 1. So just a symbol, meaning we're going to represent it as a string that has only one thing in it. All right. So our next question that we're going to look at is, we now get to look at complementing a strand of DNA. So this was actually our first question that we had for the platform. And then I realized we probably should have a couple leading into this, because this is not a hard problem to conceptualize, but it also can be a little tricky to implement. And we're going to see why that is. And it's something that if you've joined us for Programming for Lovers, we solve it there too, because it is kind of, if you're teaching somebody anything influenced by biology and programming, you probably need to solve this question. The other reason why we always solve it is it's indicative of some teachable moments, should I say, with how to program effectively, whether you're in Python or something else. So this is getting at, what's our question? It's that we have the double helix structure. We know that C binds with G and A with T, but the 2 strands of DNA in the double helix run in opposing directions. And so as a result, when you read out DNA, you want to take the complementary nucleotide. If you're looking at, I know one strand, what's the other strand? We want to take the complementary nucleotide at each position, but we also always reverse the string as well. So you might want to try and complement this string by saying TTTT, GGG, CCA. But because the strands are running in opposing directions, you start at the end of this string and then start complementing nucleotides. So we go in TGGs, complements become ACC, and the string of A's at the start of this string becomes a string of T's at the end of this string. So here's an opportunity for us to think through how we're going to solve it. I mean, I think that the doc string and thinking through the types, it's going to be very straightforward. We're going to say, I've got a function that I want to write. It's going to take a string that I'm going to call DNA. It's going to return a string too, right? And then in the meantime, what is it going to do? It finds the reverse complement of a DNA string. And our parameters or arguments are going to be DNA is a string of A, C, G, and T. And the return here is going to be a string that is the reverse complement of the given DNA string. Okay. So we know what this function is going to do on a high level. I take a string, I return a string. So that makes it straightforward. But the question is how to approach solving this question. And my question for you is, and I would say if you've seen how we approach this question in our programming course, maybe don't say anything, but my question for you is, how is it that you would come at solving this question? So here's an opportunity for you. Let me know in the chat, how is it that you would go about, in the level of code, solving this question? And you can think too, this can be motivated by how you would solve the question if you were doing it by hand, right? So we know the reverse complement of this string is going to be A, C, C, G, 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 T, T, T. But how do you think we should solve this when we take this idea and convert it into code? What could we do? And I'm not necessarily asking you for code, but what can we do on a high level? Begin from the last character? Okay. Sure. Then do what? What do we do? After we start from the last character, what do we do? Okay. Go backwards. I like this. So the ideas that I'm seeing seem to be reflected in how you might solve this question by hand. Go backwards. By go backwards, I'm going to say range backwards through the string for each new symbol. Figure out what its complement is and append it. And I should keep in mind, maybe we have the same concern that we had before where we actually need to make like a list of strings. Figure out what the complement is of the current symbol and append it to a growing string, okay, or list. All right, fair enough. So far, so good. And that's good because that's how a human would solve this, I think. And what we're able to see is, and I think many people might teach this particular approach, but what I don't see is the way I'm going to solve it. And I see, Amber, you've got an idea. You're going to start at the beginning of S and range this way, and then build the other string backwards, which is fine. That's equally valid, right? What it means is, instead of your ranging is going to be easy, I'm going to go forward to the end. but appending or concatenating is going to be a challenge. All these ideas are good. They're all very human ideas. And they're all not the way that we want to write this function. So the better way is to be lazy. Okay. And when I say we want to be lazy, what I mean is I have two different things that I'm doing. I'm reversing the string, right? And I'm also taking the complement of each particular letter that I see. So I could say that the reverse is the reverse of S, and then the complement is taking the complement of the reverse, meaning, so reverse the symbols, and this finds the complement of each letter without doing the reverse. So what this will make the coding process both easier for me to do, but also easier for me to understand, because I've taken one task and I've split it into two tasks, and each of those tasks is easier than my original task. There's a subtle point too as well, which is that if my function is wrong, if your code is just to write this function all in one, and then it breaks, well, you don't necessarily immediately know where to look. Whereas if I split this into two functions and it breaks, well, I know I have to have it bug in one or both of the functions, but then I can start testing each of these functions individually. So for that reason, I'm going to think about this process in this way. And there's a one-line way of writing this. I'm just pointing out how I've done it this way. But really what we're going to do is we're going to reverse the string, and then we're going to complement it, or vice versa. And you can do these functions in either order. And so my laziness here is a virtue, because I will complement the string, and then I will reverse the result. And that might not look, if you're still somewhat new to programming, that might not look comfortable, in which case, write it this way, I don't mind. Okay, but for now, I like thinking about this as I have a machine that's going to take in my DNA string, and it's not going to reverse anything, but it is going to find the complementary letters, and then that's going to produce a new string, and then I'm going to plug that into a new machine. This is how we think about functions in terms of inputs and outputs. This new machine reverse is just going to flip it. And so that's going to take the two tasks that I care about, reversing and complementing, and separate them out into two separate functions. These functions are now going to be easier to write and easier to work with. So I can write these in any order that I want, but I could write DNA is going to be the input string for a complement function. And I should probably write a very precise doc string, but I'm just going to say what this does is it finds the complementary strand of a given string without reversing it. And I can fill in the arguments and so forth later. But for now, what I'll do is say, here I need to apply this rule that I've seen previously, which if we look at RNA, remember, I've got a characters list, and I'm going to return that of join. So I will start with characters being a list of strings that's empty. And when we look at this character list at the end of that, that's what I'm going to return a join of. And there's a lot of different ways of writing this. So one of the ways I could do it would be to use a dictionary. One of the ways I could do it would be to say range over the DNA string and take complements. So I'll say for all of the symbols in DNA, and then I'm just going to have a bunch of checks. So if symbol is equal to A, then I'll say characters.append a T. And else if symbol is equal to C, then I'm going to append a G. And else if symbol is equal to G, we're going to append a C. And else if symbol is equal to T, that's our last case, I'm going to append an A. And then I probably should have an else that handles error checking for me. Invalid symbol. we're not working with the DNA string if we have any other cases there. Are there better ways of writing this? Sure. We talked about one in Programming for Lovers. We see there's a way of using a dictionary to simplify this down, or you could use a match statement. There's a lot of ways of writing it, but we're going to get the string that we want at the end of this process in any of those cases. Again, what's nice about that is it means I could go now here to my result and run my code just on calling the complement function. So I don't even have to think about reverse complement. I can just call the complement function and check it out and make sure it seems to be doing okay. So here's where I'm running revc.py. And I need to make sure it has the same length. I've got a small data set, so it looks like it's working because we have TTTT, GGG, and then CC and A. So we're doing the complementarity correctly, but we're just not reversing the string. So to reverse the string, that's where we also need to write a function called reverse. And it's a more generic function than anything involving a DNA strand. So we're going to have a reverse string. It's going to take a string S. It's going to return a string. And we're going to say reverse a given input string. So we can apply the same idea. I'm going to create a characters list. It's a list of strings, and it's empty. At the end of this, I'm going to return a join of that thing. And in the meantime, I'm going to say range over the string. And here's where we have something that we can think about, right? Which is, do I range over the string forward or do I range over it backward? And there is no, I just have to say, there's no preference to which direction you go. I don't think one is better than the other. I'm going to range through it forwards. And when I range over the string, I want to append. And by range over the string, I want to range over S. And consult. Actually, let's range over S kind of backwards. And append the current symbol to the growing list. So I'll say, let's make things simple. I'm going to say, what's the length of S? I'm going to be ranging over that N. Or I could just range over the length of S. But nothing wrong with writing it as 2 lines. And then the idea is, as I go through it, I'm appending something. And here is something that can be a sticking point for people who come to programming for the first time and realize, oh no, it's all math. Because programming requires noticing patterns and these sorts of things. And in this particular case, we have to think about, well, as I range up through 0 up to n-1, where n is the length of the S, and range of n, that's how this is working. So this I will range between 0, but not including n. So it's going to go up to n-1 inclusively. And our current symbol that we're looking at, so I want to append something to characters. The starting symbol that I want to look at is the last symbol. So if I'm reversing this string, then I care about appending this T. And then the next thing is going to be the G. So when I is 0, I want to grab this symbol. So I should say, it really doesn't like having a bunch of question marks. Okay, when I is 0, we append S of n-1. When I is equal to 1, we append S of n-2. When I is equal to 2, we append S of n-3, and so on. And so we see that there's a pattern here, which is that every time this increases by 1, this decreases by 1. So the pattern that I care about, which I can write out, is characters.append of S of, I take n-1, and then I subtract I. And you can see that this matches the pattern. So there's nothing wrong with writing out what the pattern is going to be. I think a lot of times when people start programming, they think there's something that makes them like less able if you write this out. No, not at all. You would want to specify like what are your individual cases. And here we say, well, the pattern I notice when I is 0, I look at this symbol. When I is 1, it's this one. So it's n minus 1 minus an extra value. And then note that this is n minus 1 minus 2. So the pattern appears to be preserved. And now I can sort of remove that because that was just a note to myself. And once I do that, I can check. So I can also check what happens when I reverse the string. And so hopefully we should see TGG, CCC, AAAA, which is what we see. We need to make sure it has the right length and so on. Okay, so I've split this into 2 challenges. Each of those tasks is pretty independent of the other. And then it's just a matter of saying, I want to combine them. But combining them is now easy because I reverse the string and then I complement it. So I have 2 working ideas, which is complementing the string and then reversing it. And now when I change this to reverse complement and save it and run it, then we're going to see, hopefully, the reverse complement of this, which is ACC, GGG, TTT, and that's good. And is there a one-liner? Sure. Would you learn anything by me showing it to you? No, you'd be like, wow, this guy's so smart. He's so good at Python one-liners. But is he a good programmer? I don't know. All right. So we should probably solve it. And we're going to say, reload the page, make sure we can solve it. We're working with strings, so there's no fancy output here. That's nice. I'm going to get my data set and copy that in. And I need to go to main and add that to what I do. And run it. And copy my string. And submit. Ooh, I don't want to do that. Oh, I got the wrong answer. What did I do? Now I've got to wait 54 seconds. All right. I think I probably truncated it or something when I copied it in, but I don't think we have the wrong answer. Yeah, I did. I got hasty. So I need to make sure that I copy the entire string next time. I think we're going to be all right. And I know we're going to be all right because I tested the function on a small data set. And I can see that the 2 subroutines that I called are okay too. But I just need to be careful next time. What questions, comments, concerns? Everybody good? I'm going to make another attempt. Let's try this again. Download data set. I'm not going to rush it. Okay, I've got the whole string copied. That was weird. Whole string copied. Copied into S. That looks good. Save. Up, run. And now I'm going to make, ooh, I don't want all those extra spaces. Make sure I get my string. And copy it in. And as you can see, a better way of doing this would be to just write it to a file. Do I want to do that today? No, but is it on the horizon? Very much so. And now we've got the problem solved. Good. Okay. And what's nice is once I unlock this by solving it correctly, I can see other people's solutions. I can check out an explanation, all this good stuff. And see what other folks do. Yeah. People love the one-liner stuff. So here's where we get really fast. There's actually a very deep point to be made about reverse complement, which is that this is a good way, objectively good way of writing this function. It separates everything out into 2 separate pieces. That makes for code that's understandable. It makes for code that's easy to test and debug. It's also not the fastest. The fastest way of doing this is to directly start at the back and range through and take the complement of each nucleotide. So notice that I have to do 2 ranges over all of the symbols. And so my code is going to be slower by probably about a factor of 2 than a faster approach like this. This is inherently related. So there's an idea here, which is that code that tends to be fast is not necessarily modular, meaning that we don't separate it out into these easy to understand digestible components. And yet, there's a principle that modularity is good. This is actually tied to a biological idea, which is that our bodies have also evolved modularity. So I have 2 limbs here, and then my limbs end in digits. Each of those can perform sort of an independent task. It's not necessarily the case that that's optimal for anything. And when people looked at evolutionary models, a lot of times what they noticed was modularity would not arise from these models. The most evolved organism of sorts would be one that was non-modular, much like the fastest solution to this problem is one that does not split this into 2 pieces. And one of the things we talk about in Great Ideas in Comp Bio in my own teaching is the first model that was able to actually show why modularity would evolve and what are the contexts that mean that modularity is a good idea from an evolutionary perspective. And it's a good idea from a coding perspective, but not if you're speed maxing. So if you're speed maxing and that's the only thing that you care about, then you get stuff like this, right? Fair enough. But I look at this and think, do I understand this? Maybe if I stare at it long enough. Whereas if I look at this, I think from a human perspective, this code is just so much easier to work with. Is it twice as slow? Sure. But it's easier to work with. It's easier to understand exactly what it's doing. It splits it into 2 easy pieces, and it solves each of those constituently. All right. Great question about, where is it that readability in code matters? What I've found is typically the more advanced someone is, the more they care about stuff like this. And what has been refreshing is to see a lot of the times when you ask AI to solve problems, a lot of the times it gives you something and that might be just because it's being trained on data, but a lot of times it gives you something that's actually pretty readable and well written from what somebody who's an expert engineer would say are good practices. So that's probably all I would comment on today with that. It's a great question. But in general, for this stream, you're going to see stuff that tends to work more with the readability side of things, work more towards modularity than towards me just trying to find the fastest imaginable solution, which if we asked AI to do that, it could do all of this in 5 seconds and then there would be no stream. So, but the modularity allows us to understand how to solve problems, I think, a little bit better. So it has good transferable value in that sense. Excellent. All right. Should we do another? Let's do another. So, and a lot of these questions that we're going to solve at this point are going to apply these same sorts of ideas. So I'll probably move a little bit faster as we go through the remaining questions that we have today. And, let us know if you have questions as we go through, chat, all that good stuff. And in the meantime, the next question I want to look at is, and I'm going to refresh my pages just so that we don't get that we're locked out. So the next question I want to look at is something called Hamming distance, which is as we go through this stream, and if you take, look at our Great Ideas in Comp Bio course 2, a fundamental notion in biological data analysis is I've got 2 genes or 2 strings or something like that. How different are they? And a very simple way of measuring that, if they happen to have the same length, and this is flawed for this particular reason, but this distance nevertheless winds up popping up in a lot of contexts, especially when we look at things that are going to be too hard to solve with perfect approaches, is the idea of just lining up the symbols and counting up the number of mismatches. So there's nothing wrong with seeing this simple approach first, because it winds up being quite useful. But if you have these 2 strings and you line them up, if they have the same length, you can count up the number of columns where they mismatch. So G against a C, G against a T, and so on. And you get a value of 7. So we call this the Hamming distance. And the Hamming distance is in this hamm.py. So we want to write this function that computes this distance, that lines these strings up and ensures that they have the same length. And then if they do, counts the number of mismatches. So this Hamming distance file is going to, or function is going to take two strings. I'm going to call them S and T, and it's going to return an integer. So I'm going to say computes the Hamming distance between two input strings by summing the number of positions where corresponding symbols differ. Okay. And the first thing I want to check then is to say, if these two strings don't have the same length, this might save us a big headache later. Let's produce an error and say that the input strings don't have the same length. And if we make it here, that's good. So it's never wrong. I mean, that took me 4 seconds, and it potentially saved us from a major concern later, right? In the meantime, now I know they have the same length. So I'm going to declare what I care about, which is a distance. It needs to start at 0. Eventually, that's what I'm going to return. And now it's just a matter of basically getting to practice the same sort of ideas that we've seen. So range over the string. Check if the corresponding symbols mismatch. So we're going to have for I in range over the length of the string. And we're just going to check. Is it the case that the current symbols mismatch? What are the current symbols? They're S of I and they're T of I. If they mismatch, what will I do? I'll add 1 to distance, and then otherwise, take no action. So this should be a quick question. I've got my S and T, and when I run it here, what we'll see is Python 3, ham.py, and we get a value of 7, and that matches what we saw on the website. So for this one, I'm just going to download my data set. I'm going to open it up. Now I need to be careful because I've got 2 strings. And then I'm going to copy those in here. And I need to get my other data set. So I've got a second string that we're going to have. And I'll copy it in here. I'm going to save. I'm going to run it. And it gives me a value of 474. So now I've just got a simple small little integer, which I can submit, and we get a correct answer. So that one was quick, which is good. We have a few to go. And I might actually save our last problem, I think, for next time. So we have one on recurrence relations, because there is an entire collection of problems on the site that are based off of a concept called dynamic programming. And this question is not so much around string algorithms. So we'll probably just cover 3 more questions here and wrap up for today, which I think that's going to give us plenty to have done. And then we'll save this question about recurrence relations for a later stream when I can just dig into, talk a lot more about how dynamic programming works, how recurrence relations work. And then we can focus in on that specifically. So this is also not related to strings. So it makes sense for us to talk through the remaining string questions. So the remaining string questions are, where does one string live in a longer string? So this is a pattern and this is a text. What are the positions where this lives in here? We're also going to look at this process of this central dogma of molecular biology tells us that DNA is transcribed into RNA, and then that RNA is used for production of a protein, often. And that process relies on this RNA codon table that converts triplets of RNA nucleotides into single amino acids. So the single amino acid alphabet is the Latin alphabet that doesn't include B, J, O, U, X, and Z. So it has 20 symbols. These are not allowed. And then our final question is going to be about, here's a bunch of strings. We're going to give you a collection of strings. Identify which one has the highest GC content. So this is a fancier version of the question that would just ask, what is the GC content of a string? Meaning, what's the percentage over the total number of nucleotides? What's the percentage of G's and C's compared to the total? So we have 3 more string problems to wrap up with, and we'll do these one at a time. The first question I'm going to take a look at is this finding a motif in DNA question. And this is a fancy way of saying, where is it that one string occurs exactly? We could modify this problem by saying we could also count mismatches, but where is it that this pattern occurs exactly in a longer string? So our input is going to be 2 DNA strings, of course. They don't have to be DNA strings necessarily, so I'm going to write this as a general function to just find the number of matches of a pattern in a longer text. And we want to find those locations. So this helps us think through what those locations should look like. This is essentially just a list of integers. And there's a little bit of a note, I should say here too, which is if we look at the sample data set, most languages are what are called zero indexed, meaning if you say, what is the starting position AT? Well, we would normally say position 1. So the G is at position 0, and this AT is at position 1. The next occurrence, which overlaps with that occurrence, would be at position 3, 0, 1, 2, 3. And then the final occurrence would be at position 9. You can count that up. We made the fated decision on Rosalind that a lot of the sample outputs would be 1-based. So this is saying that we're counting it as a human, 1, 2, and so on. So this occurs at position 2, this is occurring at position 4, and then the final occurrence would be at position 10. And so unfortunately, what we're going to have to do is make sure that when we print this, we add a 1 to our result. So I'm just saying that now, so when we move to code, and this is going to be in subs.py, we're going to write this function called find substring positions. And it's going to take a string and another string. And I even could get a little fancier with my variable naming and call it pattern is the first string and text is the second. And then what I want to return is a list, and that list is just going to be a list of integers. So I'll write, again, an abbreviated documentation string to say what this does. It finds all starting positions of a pattern within a longer text string. Okay. And I'm going to say this is 0-based. So I declare what I care about. I've got a list of integers, and that list of integers is empty to start off with. It may always be empty. At the end, I'm going to return it because I might not find anything. And that's why we use a list, because a list is mutable, and we don't know exactly how many occurrences we're going to see in advance. I also could do some checking. So one thing we might do is to check that, I don't know, the length of the pattern is not bigger or is smaller than or equal to the length of the text, etc. So think through, as I go through this, you might think about what are the other sorts of things that you might want to ensure about the data set. I could just say if the length of the pattern is greater than the length of the text. Well, you could do a couple of things. I might raise an error. And say it's too long. Or I could just return an empty list. So there's lots of choices that I could make there. I'm more interested in focusing on solving the question for now. And when I'm solving the question, what I'll say is, let me declare a couple of variables to help me. So n is the length of the text. And let's say k is the length of my pattern. And here's where I get an opportunity to use a very standard fact that every computational biologist who works with anything associated with strings is going to know. This is a primary comp bio fact. If there are stone tablets of comp bio and there's 10 things you could write down, this is probably one of them, which is a string of length n has n minus k plus 1 substrings of length k. And they range from the starting positions 0 to n minus k. That means that I can solve this by saying, let's range our starting positions from 0 up to n minus k plus 1. When we do Python ranging, we don't include this second element. So we go up to n minus k plus 1 not inclusively. And that's right because we want to go up to and including n minus k. We also don't need the 0 in Python because that's included by default if we don't include it. And now we say, is the current, you know what, I want to write this. I'm going to put text first. I think that's how my test works. So you just need to establish in advance the order, but let's put text first in our inputs. And I'll say, is the current substring equal to pattern? And if it is, well, I want to append it to the positions. So I'll say, how do I access that current substring? I'm going to say, is it the case that text of something equal to pattern? And if it is, well, here's where it really doesn't like me putting multiple question marks in. I'm just going to put X for a moment. And if it is, here's where I'm going to say positions.append the current starting position that I'm on. That current starting position is called I. Now, the only thing that's a challenge here is to fill in X. So here's where we need to say, what is the current substring? So the current substring starts at position I, and it has length k. So that's all you really need to know. And I'll show you a trick that's included in a lot of languages, but it's the Python standard, which is that in Python, when you have an initial index and then a final index, the length of the string that you care about, the length of this substring, is going to be y minus x. It's a great thing to know. Because what this means is, this is the substring starting at position x that goes up to y, but it doesn't include y. And so, if for example this were 10 and this were 17, you'd get the substring going from position 10 up to, but not including position 17. You say, why would you do it that way? Well, this is the same way that ranging works. But it means that you get an immediate answer for the length of this string. So this string would automatically, it's going to have length 7, and the reason why is because you subtract the smaller number, the lower index, from the higher index, and that gives you the length. So that's very convenient, because a lot of times when I see people come to programming, this can be a challenge. But what's nice about it is I always think about this first element is the starting position in the substring. The second element is just the starting position plus the length of the substring that you care about. So all I have to do is say my starting position here is I. My ending position is I plus the length, and then you're done. So it is that easy if you think about it in that way. The substring is defined by where it starts and what its total length is. Now when I save it, I actually should be able to check my sample data set. So we've got find starting substring positions of S and T. S here is the longer text, T is the shorter pattern. So we've got our order of our inputs is correct. And I can run this by saying Python 3 subs.py, and we see 1, 3, and 9. We look at the sample output, and there's two issues here. Number one, I need to make sure that I add 1 to it because we're converting from 0-based to 1-based. I'm going to keep my solution the same because I've solved this correctly in Python. It's fine for the solution to be the same. What I'm going to change is the printing. So the printing needs to get rid of the comma and the brackets, and I need to do a little bit of a better job there. But then I'm also going to add 1 when I print each of these values. So I'm going to say for each of the positions that we see, so I'll range over this. It's giving us a list of integers. When I range over it, I'm going to say print the position, and then I'm going to say +1. And we'll see that that works, but the problem becomes everybody's going to go on its own line. And here's where we're going to use the fact that in Python, there is a built-in extra parameter to print, which if you don't specify it, is going to place everything on a new line. And if you don't want everything on a new line, you simply need to specify a different end. So after I print each of these things, I'm not going to print a new line. I'm instead going to print a space. And when we run it, there you see we have 2, 4, and 10. And that 2, 4, and 10 is going to match what we see here. So everything should be good. And now I should be ready to download a new data set, take a look at what it is. Here's my text and my pattern. If I look at my data set is this thing. Okay. And then I'm going to run, and I get this list of positions, and I can copy that back in. And click submit. And we get that right. So we have 2 more that we want to address today. We're going to look at this process of translating RNA to protein, and then I'm going to look at our GHC content question. How are we feeling about all this? The chat's been quiet. Had some good initial questions. Give me a 0 to 10 about how comfortable you are with all this, just so I know. Or maybe where you started and where you are now. after an hour, a little over an hour. Feel 10. Okay, good. All right, stick around. That's good. I'm glad we got some 10s. Maybe you're just being nice. Wherever your position is, I guarantee you, unless you're one of our top 100 solvers, we're going to have a problem that's going to wind up being challenging. So if you're 10 now, that's fine. 11, that's good. How about 27? It's one of the things we're proud of with the site, though. We start with these challenges that are important, but also, I think, beginner level types of challenges. And then the next thing you know, as you go through this, you start to have things that are pretty complicated. So we'll get there. All right, let's do 2 more problems. Great. So our next problem is going to look at taking an RNA string and applying the genetic code, central dogma of molecular biology, to convert it into an amino acid string. And this is based off of this process of the ribosome facilitating this translation process using transfer RNAs that take a messenger RNA molecule and produce a peptide strand of amino acids from it. And the critical observation here, which took years of biological research that at that point in time was amazing, was to prove that this process takes triplets of nucleotides and converts them into single amino acids. So as a result, we wind up with a table that tells us precisely what each triplet or codon corresponds to as a single amino acid, and some of these corresponding to no amino acid, meaning there is no transfer RNA that has a corresponding amino acid to convert UAA. And if that happens, then the translation process halts. So as a computational challenge, the question becomes, well, if we have an RNA string, can we translate it 3 letters at a time until such a time that we encounter a stop codon, or until such a time that we encounter the end of the string? This is in PROT.py, and you'll notice that I've tried to be nice and give us this codon table as a constant dictionary mapping strings to strings at the start. So what we want to do is add our translate function. And our translate function is going to take a string as an input, and let's call it RNA. And it's going to produce a string as an output. Okay, and we could specify a lengthy docstring here. Again, I'm going to do a slightly shortened one, which says that it translates an RNA string into an amino acid string, 3 nucleotides at a time, and stops at the end of the string or the first stop codon encountered. And there's a lot to check here. Again, we could add some error checking and make sure that something is actually, every symbol in the string is contained in the RNA alphabet. So the last thing we want to do is allow a letter that's in the string that's outside of that. Okay, so I'll say good idea. Check that every symbol in RNA is an A, C, G, or T. And I've realized I need to call this DNA, of course, because our sample data set is U's. Okay, so our sample data set contains these RNA strings. So I don't know why I thought this was DNA initially, so this should be a U. I could have sworn I saw a T up here. All right, so everything's all right. So we need to check that. I'll leave that to you. So this would be like arranging over the string to make sure that everything is okay. For now, I think what's fair about a lot of the Rosalind problems is that we don't have to focus on that sort of thing. So I'm going to have the protein symbols that we have is going to be a list of strings, and it's going to be empty. And then way down here, I'm going to return the join of them. So we're getting to use that idea that we saw before, which is instead of a bunch of string concatenations, we're going to form a list of symbols, and then I'm going to join them all together into a string at the end. Now we want to range over RNA 3 symbols at a time, converting the current one to an amino acid and appending it to our list. What's nice is that Python provides a very straightforward built-in way of doing that. So we're used to seeing for I in range of 0 up to the length of the RNA. But what I would like to do is skip 3 symbols at a time, because what I want to be able to do is say codon, the current codon, is equal to the substring starting at position I of length 3. We get to see the same thing that we just saw. If I want a length 3 substring, I just add 3 to the bottom index. And when I range over the string, I want to start with AUG, and then I want to jump with to GCC, and then I want to jump to AUG. So that means I want to skip not 1 symbol at a time when I do my ranging, but I want to do 3 at a time. Python allows this with a third index to the range function that will tell us what to add to I every time through. So this tells us to add 3 to I. So we start I at 0, then we go to 3, 6, and so forth. Once I have that codon now, I'm going to convert it to a protein symbol, so an amino acid character, by saying that the current amino acid is codon table of my codon. This is a dictionary mapping strings, triplets, to single amino acids, which is what we wanted. And then once I've done that, I can say, well, notice that there are certain stop codons. How are those represented? The stop codons are just empty strings. We could have encoded it as a special character or something like that. We just have it as an empty string. So if we just have it as an empty string, then one of the things that we might do is to say, when we're here, say, is it the case that the amino acid is just empty? So this means we're at a stop codon. And if that's the case, well, I want to make sure that I stop the ranging process. So here's where we're going to encounter the keyword break. So this will stop the outer for loop. And then at that point in time, we're just going to go ahead and return the join of our list of symbols. If it's not an empty string, we could have an else. And I'll say protein symbols dot append the current amino acid. So keep creating this chain of amino acids that we're going to return. Now, technically, there's a little bit of a way to improve this, right? Which is to say, I don't need the else here, because if I enter the if statement, then I've stopped the for loop. So if this is not true, I'm just going to write this function in this way. Okay, so if we make it here, we haven't broken, so we know that amino acid is not empty. And then we can return it. So this amazing, complex biological process on the level of code just gets converted into a pretty short function for translation. And I can save it. And if I can pull the ribbon up, I can run this by saying Python 3 prot.py. And that gives us our mama protein string, which is what we see here, which is good. So we don't have any indexing issues or anything. We need to make sure the where this could break would be the start or the end, but we're getting all those symbols. I've got a current data set, and with my current data set, I'm going to copy this in. It's just a single string. Hopefully everything is highlighted, and then I will run it. And when I save that and run, I get a really long amino acid string, so I want to make sure that I select the whole thing. Next time we're probably going to have some built-in code for writing to a file, but I wanted to have the simplest explanation here. Yeah, I was about to make a mistake. So we're going to start here and copy everything. Okay. And when I copy that back into the platform, we're going to see that hopefully we get one more correct submission, and we do. All right. One question to go. So our final question is going to look at GC content. It's going to rely on us to be able to compute the GC content of a string, which essentially just says count up the number of G's and C's that you see in a string, and then divide it by the total. The reason why we do that is one of the ways in which you could differentiate organisms is by their GC content. It is a very simple metric, but this GC content metric varies widely across a lot of different organisms. So it's around 50% for most eukaryotes, but we also have that prokaryotes can have a lot more variation. So sometimes they have GC content substantially higher than 50%. And so if you were presented with a small strand of DNA, you could identify that it's most likely prokaryotic based off of just this simple test. This is also an important thing to consider because a lot of times algorithms that we have for biological data analysis might have a built-in assumption that the frequency of ACs, Gs, and Ts is the same. And one that I would say comes to mind off the top of my head is motif finding, where if you were looking for the same binding region upstream of a lot of genes, and you're looking for similarities there, your algorithms need to appreciate that the GC content might be not 50%. And we see this in our bioinformatics algorithms project or Great Ideas in Comp Bio, where we talk about this very thing. So the algorithm that you design that might work perfectly if it's assuming equal frequencies starts to get fooled if those frequencies become significantly skewed from everybody being equal to 25%, or where you have a GC content equal to 50%. So the way that we framed this challenge was to say, you solve this question of determining the GC content of a string, but there's a little bit more to it, because we're going to be finding of all of these, which is the one, what's the ID of the string that has the maximum GC content, and what is that GC content. So if we were to solve it, and we look at our starter code, and we look at gc.py, there's a little bit of parsing here. And so as we go through this, the parsing code will be helpful. And I have this included here to help us out a little bit by taking a string, and in particular, the string I'm going to take is this entire data set, and to convert it into a nicer form. So I'm not going to go through how this parsing code works, but the idea is this is the string corresponding to what we see on the website. And this is in FASTA format in the sense that you have an initial line with an ID of the sequence that starts with this greater than symbol. And then the sequence itself is contained on remaining lines. And each sequence or each string might be on multiple lines here. So we have a little bit of code called parse FASTA to take this string and parse it out as a dictionary that maps sequence IDs to single strings. And so we haven't written a function here, but one of the things that I might do is, well, we could print, I could comment this out for a moment, and we could print the sequence database so you see the format that it's in. So if I go to my terminal, and in this particular case, what I want to run is Python 3 gc.py, then you're going to see we're mapping the ID, which is a string, to the DNA sequence that's associated with it as a single contiguous string. Same thing here. So 5959 is this. 0808 is this. Now that we understand how the database works, that of course is going to help us figure out, well, what's the function that we're going to write? I think this is maybe, from a certain perspective, the trickiest function that we have seen thus far, because there are a fair number of moving parts. But if we always think about on the highest level, what is the problem that we want to solve? Well, I look at my output and I want to return two things. I want to return a sequence ID and I want to return its GC content. So this is almost like a key value pair. And so if I'm interested in that particular pair, what I'm interested in is a tuple of two values. So when I find the highest GC content, it's going to take my sequence database, and that's going to be a dictionary of strings to strings, mapping these IDs to the strings associated with them. And what we're going to produce is a tuple of strings and floats. And we should specify what this does. So our function takes a sequence database mapping IDs to strings as input and returns a tuple of two elements, the ID and GC content of the sequence having maximum GC content over all strings in the database. All right, pretty good. And again, I can write out specific arguments, specific outputs, but in the meantime, I can also do a bunch of checks. In the meantime, I want to say, well, I want to range over this entire database. And at the end of this process, what I want to return is the best ID that I find and the best GC content that I find. And when I do this, that means that I need to set an initial value of this best ID to an empty string, which I'll fill in, and the best GC content that I find equal to 0.0, for example, because everybody's going to have a GC content of at least 0.0. And then I want to range over the database, and I'm going to want its keys and its values. And when I do that in Python, I'm going to need a range with a for loop that says I'm interested in both the sequence ID, that's the key of my dictionary, and then the value of the dictionary is going to be, well, the sequence itself, which is a string. So it's tempting to write sequence database, but when we range over both the keys and the values of a dictionary in Python, you're going to need to get these as tuples. And to do that, you need .items at the end of your database name. And here is where we get to practice what we have seen earlier, which is I want to be lazy. And I could think about, well, now I've got to compute the GC content of every individual element that I have. But the current sequence that I have is just sequence. So this is just the string that I have. It's like one of these. So we have the sequence ID and then a string. And all I actually care about is getting its GC content. And I'm going to hand that off to one of my minions. So I'm going to say that the current GC content is computed from a subroutine called GC content. And here's where I'm going to be able to say, is that current GC content of the current string, is it better than anything that I've seen thus far? So does it beat best GC? And if so, well, update both best GC and the ID. So I'm going to say that the best GC is equal to that current GC value. And the best ID is the current sequence ID. So you'll notice that it's colored in this darker blue because I haven't used it. So now I get to make sure that I use it, and I set that sequence ID. That makes my work so much easier because the only thing now that's remaining is to write an individual subroutine called GC content that takes in my DNA string and returns its GC content. But that's going to be a very straightforward function, right? That is a much easier task than what I've already solved because I'm going to have a function called GC content. And it needs to take a DNA string as input, and it needs to return just a float. So it's going to return a decimal. And in this particular case, we can specify our documentation string to say this computes the GC content. And by GC content, I mean the percentage of G's and C's among the total of a given DNA string. Okay. Important check, which I won't do synchronously, but an important check you might want to add is to verify that we have a valid DNA string. And I want you to notice that's a check that's come up quite a lot in us solving these questions. So maybe add a helper function for this that you could call. So for example, you could say something like if not is valid DNA, and maybe that returns like a Boolean. So it ranges through the whole string and makes sure that you have a valid DNA string. And if you don't, then it's going to return false. And you'd produce an error. But for now, I'm going to assume that I have a valid DNA string because our data are good on the platform. And I'm just going to say, well, what's our GC count? It's going to be corresponding to counting the number of symbols of the current sequence DNA of a G. And notice what I'm going to do here. I'm going to pass my work to yet another subroutine. So I'm going to do the same thing for the C's. I'm going to count the number of symbols that I see in DNA that are C. And then once I have that total GC count, then I'm going to divide by the total. So what I'll do is I'll return the GC count divided by the total number of symbols in the sequence. Well, that's just length of DNA. And then I need to multiply this by 100 because I always return the GC content as a percentage. So this fraction will be between 0 and 1, and I'm going to multiply it by 100 to make sure that I'm okay. Of course, I haven't written count symbols. And we keep pushing these details down, but what is count symbols going to do? It's going to take an arbitrary string, and it's also going to take, I could have it take a single symbol. I'm going to write this in a slightly different fashion by saying this is for an arbitrary text, and it's for an arbitrary pattern within it. And so when I write that function, it's just going to return an integer. And here's an opportunity for me to note that I've actually already written a function that's very similar to this. So I'm not counting symbols. I'm counting pattern matches. Okay, and so I'm able to write an even more generic function that counts the number of pattern matches of a pattern in a text. And that's so similar to what I've already done. Like you might remember, we already have a function called find substring positions that we wrote. And the only difference here is it finds all of the positions. And I'm going to copy it in because we know how to find all of these positions of a pattern within a longer text. And if we look at our function that we wrote, all I need to do is count up the number of elements that I see in the positions that I found. So if I have that these positions are 2, 4, and 10, well, my Python function is going to return 1, 3, and 9 for this data set. But if I knew that the positions were at 1, 3, and 9, I just need to count up the total number of elements there. So if I've written this function already, I've done the hard work. And I'm going to keep this function here. I could also import it, which is something that we'll talk about soon. But I've got this function, which means that if I want to write my function count matches, what does it do? It counts the total number of occurrences of a pattern substring within a longer text. And yes, I could do things like if the length of the text or pattern is too long. Maybe I return 0, or maybe I have an error message. But in the meantime, I could say, if I want to know how many patterns occur in text, I could say, well, what are the starting positions? And I can find the substring positions of that pattern within that text. And once I know that, well, I just say, how many of them are there? And I'll return the length of starting positions. So the number of elements that I see in this list of positions is going to be the answer to my question. So I've already solved a more general problem. And this problem elucidates what I'm getting at, which is you might not have thought initially that this function that we already wrote would ever be helpful in solving this particular question, but it turns out that it is. And so our code is going to start calling itself in different places. And as we move through this, we need to get a plan for how we're going to make that process effective and how we're going to reuse code as much as I possibly can. At this point in time, though, I think I've solved the question. I hope so. So I should be able to save. And I should be able to bring up my terminal. And I want to run, in this particular case, I want to make sure that I update main. So I've printed the sequence database. What I actually want to print is find highest GC content. Okay, so I want to access the solution, and that's the solution that I care about. Make sure that I name it correctly. And when I hit up, I get Python 3, and now I want to run gc.py. And when I ran that, notice that I get the solution that I had before. And I can make this a little bit fancier if I wanted, but I'm actually only printing 2 things. So I think for the purposes of solving the question, when I look at computing the GC content, I just need to copy in the ID on one line and the GC content on another. So I've solved the solution. I guess I could print solution 0 and solution 1, and let's see what happens there. And now that gives it to us on 2 different lines, and we've got everything formatted according to the website. So it's just a matter of going to the platform, downloading a data set, opening it up, copying this into what we had before, which is our this big doc string. So having these triple quotes allows you to have a string that spans more than one line. And now that I've got that, I'm going to say, okay, I've got all these strings. They're in between triple quotes. So we've got a big string. And when I run that, here is the output that I want. I'll take it back to the platform. It finds this ID as having GC content of a little over 52%. Go back to Safari and submit it. And we've solved the problem. And we've also unlocked our string algorithms badge level 1, which is good. All right, that's good to see. So you get a little bit of a reward from the platform for all those string algorithms that we solved. That, I think, is plenty for now. We've seen quite a bit here. We started getting comfortable with working with ranging over strings, working with dictionaries that are based off of these strings and so forth. Next time we have quite a bit to do. So we've got a lot more problems to solve. I'm going to refresh where we are. Notice all these yellow problems are problems that we can now start looking at, which is exciting. So we have a lot of directions that we could go in. We could do some more string analysis. We could dip our toes into dynamic programming and recurrence relations. We could go and look at genetics questions, start working with networks. Now that we've established this foundation, we have a lot of different directions that we could go into. So that's what I'm excited about getting to do as we move through this, but we're going to save it for next time. So thank you so much, wherever you're from, for joining us today in our stream. As always, I say like and subscribe so that we stay visible. Join us on our mailing list. You can check out our description for that information, or come join us in the Discord. We probably should set up a special channel in the Discord for our Rosalind slow run. So if you have any questions on what we do, you can come join us there, ask questions. We're happy to join you. We've got other courses that we're running. Check those out. And otherwise, I'll look forward to seeing you next time. And thank you for joining me. And until I see you next, happy coding.